Welcome, my dear microbiology friends. Today, as I have already mentioned in the part one of the bacterial colonies, we will be discussing today on the part two of bacterial colonies. What we are dealing today is part two colonies, like we will be studying on the growth pattern on an agar of colonies and how to streak on an agar slant. Those are two small things we will be talking today. Growth pattern on agar is it is crucial thing to recognize a growth morphology bacterial colonies on a agar slant used for cultivation of pure cultures, and the principle is used is like we have already talked about on the first section of the bacterial colonies, that one cell will divide into 2 to 4, 4, 8, 1 million, 2 million, so on. So there is a binary fission of colony formation. Eventually a colony appears of the original cell was deposited. Once the purity of the colony has been confirmed by appropriate staining procedures, the cells can be transferred to a sterile medium to begin a pure culture. You have color, size, shape and texture of the microbial growth are determined by the genetic makeup of the organism. The genetic expression is generally influenced by the environmental factors including nutrient availability, temperature and incubation time. Colony characteristics may be viewed with the naked eye and assistance of a colony counter. Colony counter, I think the figure I had shown you in the last class. Basic characteristics, basic categories of the growth include colony shape, margin, edge, elevation, color and texture. Colony shape may be described as circular, irregular, punctiform, and margins may be entire, smooth with non-irregularities. Undulate is wavy, lobate, lobed, filamentous or rhizoid branch-like roots, and colony elevation features are flat, raised, convex, pulvinate, very convex, ambonate raised in the center, and colony texture may be moist, mucoid or dry. Pigment production is another useful characteristics, may be combined with optical properties such as opaque, translucent, shiny and dull. These are the simple things we want to know. If you have any doubts, you can repeat the video and replay it. Other things like what we want to know is how about we streak on a medium. That is a simple thought, we can just talk along with this. I'll just teach you about that. The identification process of a unknown micron relies on the obtaining a pure culture of that organism. That is the identification process. The streak method produces individual colonies on an agar plate. Portion of isolated colony is then transferred to a sterile media to start a pure culture. Microbial culture consisting of two or more species is said to be mixed culture, whereas a pure culture contains only a single species. Okay, obtaining isolation of individual species from a mixed sample is generally the first step in identifying an organism. Commonly used isolation technique is the street plate. In the street plate method of isolation, a bacterial sample, always assumed to be a mixed culture, is streaked over the surface of the plate agar medium. During streaking, the cell's density decreases, so eventually leads to individual cells being deposited separately on agar surface. Cells that have been sufficiently isolated grow into colonies consisting only of original cell types. Because some colonies form from individual cells and others from pairs, chains, clusters of cells, the term colony forming units is the more correct description of colony origin. The common streaking technique is the quadrant method, which uses the 4-streak pattern. I will show you the figure. It will be shown, and that is the common method, streaking isolation on a quadrant method. I will try to show you a video of that, possibly. That's all on the bacterial colony features. For details, you can refer Tope and Wilson and any other books. Thanks for listening. Thanks for watching. Dr. Jeevan.